Hello, this is the second part of vaccine overview chapter, and in this lecture, we will learn about DNA vaccine and then RNA vaccine. So, first, DNA vaccine. So, the strategy for DNA vaccine, I mean, the first few step to generate DNA vaccine is almost same like the recombinant vaccine. Okay, however, I mean, in this case, such as I mean, if you want to develop a DNA vaccine, so first you have to locate or get the genes which encode the desired antigen. Then we have to clone this gene into a plasmid. Then we'll use this plasmid as the vaccine. Okay, so first isolate the gene which will encode your desired antigen, insert it into plasmid, then use this plasmid as a vaccine. Okay, so after you prepared your plasmid, then I mean that is the vaccine. So then it is injected into the muscle of the animal being vaccinated. So usually, I mean, so we can administer the DNA vaccine into the animal with a gene gun by bombarding the skin with DNA coated gold particles or by nose drops. Okay, till date there is no vaccine, DNA vaccine approved for human use. However, one vaccine, it is again West Nile vaccine, is approved for animal use. Okay, so it is as a animal vaccine against the West Nile virus. So this photo, it depicts that strategy. Okay, so the West Nile virus is a RNA virus. Okay, so it's not a DNA virus anyway. So first you have to locate the RNA, okay, that encode the desired antigen. Then, okay, so then we can we can convert the RNA into DNA. So you know that, I mean, by using PCR, but in this case we have to use reverse transcriptase. Okay, so using reverse transcriptase we can convert RNA into DNA. Okay, so first locate the RNA that encode the desired antigen, then uh, make it DNA by reverse transcriptase. Okay, then by PCR amplify the gene and then insert or clone the gene into the plasmid. So we already the vaccine, and this is the vaccine. Actually, this cloned plasmid is the vaccine. So then once the plasmid is prepared, then we have to increase the number. So how the number is increased? Okay, basically it is done first. You need to insert this plasmid into your microorganism. So usually E. coli is a good, I mean, there are some commercial strain of E. coli, and those E. coli are good to increase the number of plasmid. Okay, they are sometimes called high throughput E. coli. Okay, so once you get the plasmid, that is the DNA vaccine, then it is transformed into the E. coli, and then the E. coli is cultivated in large quantity. So when the E. coli will grow, they will also produce this. I mean, when the E. coli will multiply, definitely the plasmid will be multiplied. Okay, so cultivate the E. coli in large quantity, and then harvest the E. coli cell and extract the plasmid and purify from them. Okay, so then this plasmid can be used as the these plasmids are used as the DNA vaccine. Okay, so the DNA vaccine also has some advantages and disadvantages. So advantages are DNA is very stable. Okay, so it resists extreme temperature and hence storage and transport are easy. Okay, and a DNA sequence can be changed easily in the laboratory. Okay, so it's kind of easier to I mean, change or modify in the DNA sequence. Okay, and the inserted DNA does not replicate and encodes only the proteins of interest. Okay, so I mean, the DNA definitely it will not replicate. Okay, so there is no chance it will cause the disease. Okay, and this DNA will replicate only the only our desired protein that we are going to use as antigen, that is vaccine. Okay, so however, the DNA vaccines, they also have some disadvantages. One is potential integration of DNA into the host genome, leading to insertional mutagenesis. Okay, so I mean, in case of DNA vaccine, so, okay, so the DNA first need to be in the nucleus. Okay, because I mean, when a protein is produced from a DNA, first the DNA the first mRNA is produced from the DNA. Okay, and this happens in the nucleus. So the DNA first it need to be in the nucleus. So as it is in the nucleus, so there is a chance that the DNA they can get integrated into the host genome there, which will cause insertional mutagenesis. Okay, and another disadvantage is induction of autoimmune responses. So anti-DNA antibody can be produced, okay, against the introduced DNA. Okay, so it will definitely cause autoimmune response. Okay, so that was about DNA vaccine. Now RNA vaccine. RNA vaccine, as in the DNA vaccine, the DNA is used as a vaccine. In case of RNA vaccine, the messenger RNA strand that encodes for a disease specific antigen is used as vaccine. That means in case of RNA vaccine, messenger RNA is used as vaccine candidate. Okay, so what happens, once the in this kind of vaccine, RNA vaccine, once the messenger RNA strand in the vaccine is inside the body cell, then the cells use the genetic information to produce the antigen. Okay, and this antigen is then displayed on the cell surface where it is recognized by the immune system. This is the overall mechanism by which the the RNA vaccine work. Okay, so I will explain it through a figure. So you all heard about the, I think most of you already know about the uh, Moderna. Okay, so Moderna is a company, biotech company. They are developing a vaccine against the COVID-19. Okay, so they are all already in the phase 3 trials. So I hope soon we'll get the vaccine. Anyway, so this photo is depicts the approach or a strategy by which the RNA vaccine works. Okay, so I'll try to explain. So first, say they develop the vaccine against COVID-19. So first, they isolated the messenger RNA, which induce, which produce the, which codes the spike protein of the coronavirus, no, COVID-19 virus. Okay, so they isolated that messenger RNA and then coated it with lipid nanoparticle. Okay, and then injected this. So they isolated the messenger RNA that encode the spike protein of the COVID-19. Okay, then they enclosed it or covered it, this messenger RNA, with lipid nanoparticle. Okay, so this is the vaccine. So then they injected it into the muscle of the host. Okay, so once the this vaccine is in the muscle of the host, so what happens? Okay, the muscle cells, okay, the muscle cells, they take this vaccine. That means the messenger RNA, it goes inside the cell, muscle cell, and there is ribosome. So we know that the ribosome, they read messenger RNA and produce the protein. So in this case, the ribosome, they will read this viral messenger RNA and they will produce the viral protein. Okay, protein chain. And this viral protein chain, and some of this viral protein chain, they can present it on the surface of the muscle cell. Okay, and can act as viral antigen. Okay, and some of the viral protein, okay, they will get processed, okay, and will be present in the muscle cell surface via MHC1, major histocompatibility complex 1. Okay, so for this, as the viral protein, there, that is the antigens, they are presented in the surface of the muscle cells through MHC1, so they can attract the T helper cells, and the T helper cells will come and induce vaccine, okay, against this viral protein, that means against the virus. Okay, induce, antibody. Okay, the T helper cells will come and they will ultimately induce the immune cell, okay, to produce antibody against this viral protein. Okay, so this will happens against the protein expressed by the MHC1. And but I already told you earlier that some of the viral protein, they will be presented in the cell surface. Okay, so they can directly, the antigens, they are expressed in the cell surface, they can directly activate the B cell and can induce some immediate immune response. That is, these B cells, activated B cells, will readily produce some antibody. Okay, Okay, so and not only the muscle cells. Okay, some of this RNA they will be taken by the specialized APC cells in the lymph node. That means the specialized antigen presenting cell in the lymph node. Okay, so once okay, so the lymph node, okay, they can either, I mean, actually two things will occur simultaneously in this case. One is the APC cell, they will take the messenger RNA. Okay, and another things can happen, they can directly take the exogenous viral antigen. So from where, what is the source of this exogenous viral antigen? Actually in the muscle cells. Okay, in case of muscle cells, some of the viral antigens they will be presented in the cell surface. Also some viral antigens they will be excreted from the muscle cells, okay, in the stream. So that those can and those are the exogenous viral antigens. So the APC cell, they can both take the, they will take both the messenger RNA, that is the virus, that's the vaccine, and also this exogenous viral antigen. Okay, so what will happen once the messenger RNA is inside the cell, the ribosome will produce viral protein from those messenger RNA. Okay, and then those viral protein, I mean newly synthesized viral protein, will be processed and ultimately presented in the cell surface by MHC2. Okay, so actually kind of similar thing will happen for the exogenous antigens. So once the exogenous antigens they are inside the cell, they will get processed and presented by the MHC2. Okay, so only difference between these two is in case of exogenous antigen, lysozyme will process them, okay, will digest them, but in case of viral protein chain, okay, produced by the ribosome, they will be processed or lysed by proteolytic vesicle. Okay, anyway, in either case they will be presented in the cell surface of the antigen presenting cells. So once the antigens are presented against, the T helper cells will get attracted, okay, and bind with this, with the MHC2, I mean that, okay, and then they will activate the B cells, okay, to produce, to get differentiated into plasma cell and memory cell. Okay, so these are the basic steps or mechanism by which a mRNA vaccine work. Okay, so this is the strategy of Moderna vaccine. Okay, anyway, so okay, so this was, okay, this is one strategy or mechanism of RNA vaccine. Actually another form of RNA vaccine is also possible. So in that form, I mean those mRNA vaccination is one in which the mRNA encodes for a fully human IgG antibody. Okay, so I mean in first strategy the mRNA encode the antigen, but in there is another strategy. In that strategy the mRNA it's and don't encode the antigen, rather it encodes the antibody. Okay, so in this form the messenger RNA codes for antibodies that are identical or resembling those of the antibodies found in a patient with prior history of potent immunity. So for example, you want to develop a vaccine against the COVID-19 using this strategy. So in this case you actually have to get the gene that encodes this, encode the antibody against the COVID-19. Okay, so if you have that, I mean actually you need to have the messenger RNA that encode the COVID-19 antibody. So if you have that messenger RNA, then you can use this messenger RNA to make your RNA vaccine. Okay, so this is another type of vaccine. The Moderna vaccine is based on this strategy, not on this strategy. Okay, basically the, you know, the another company, the Oxford, there, I mean the Oxford, they are also their vaccine is also promising and now is in third phase. It's also a messenger RNA vaccine. Okay, but the difference is the Moderna, they use lipid nanoprotein to coat to make the vaccine, but the Oxford group, okay, they use adenovirus as a carrier or vector to make the messenger RNA virus. Okay, so they also use the messenger RNA, but then they insert the messenger RNA into adenovirus and use that adenovirus as a vaccine. Okay, anyway, so there are different types of RNA vaccine, usually three types: non-replicating messenger RNA, in vivo self-replicating messenger RNA, in vitro dendritic cell non-replicating messenger RNA vaccine. Okay, so the non-replicating messenger RNA, the simplest type of RNA vaccine. Okay, and a messenger RNA strand is packaged and delivered to the body, where it is taken up by the body cells to make the antigen. Okay, non-replicating mRNA vaccine. And there are also in vivo self-replicating mRNA vaccine. Okay, so in this case, the pathogen messenger RNA strand is packaged with additional RNA strands that ensure it will be copied once the vaccine is inside a cell. Okay, so this means that greater quantities of the antigen are made from a smaller amount of vaccine, helping to ensure a more robust immune response. This strategy was applied in the Moderna vaccine. Okay, then another types of RNA vaccine, the in vitro dendritic cell non-replicating messenger RNA vaccine. Okay, so in this case, the dendritic cells are immune cells. Okay, that can present antigens on their cell surface to other types of immune cells to help stimulate an immune response. So these cells are extracted from the patient's blood, transfected with RNA vaccine, then given back to the patient to stimulate an immune vaccine reaction. Actually, I mean, in this strategy, dendritic cells are used to produce the vaccine. So how it is done? First, you need to collect the dendritic cells, okay, from the patient's blood. Then you have to transfect these dendritic cells with your messenger RNA or the RNA vaccine. Okay, then dendritic cells, they are antigen presenting cells. So upon transfected the dendritic cells, they will present the antigen in the cell surface. Then they are ready to be used as vaccine. Then you have to inject these dendritic cells back into the patient's body. Okay, actually, this same strategy is also can be used to make some groups, some scientific group, they are actually they're trying with this strategy to make vaccine against the cancer. Okay, so when we will learn about vaccine, cancer vaccine, then we'll learn about this. Okay, so anyway, unlike all the other vaccines, messenger RNA vaccines is also have some advantages and disadvantages. Okay, so first, the benefits of messenger RNA vaccines over conventional approaches. So what? First, safety. So RNA vaccines are not made with pathogen particles or inactivated pathogen, so are non-infectious, and RNA does not integrate itself into the host genome, and the RNA strand in the vaccine is degraded once the protein is made. In case of DNA vaccine, there is a chance that DNA, they can get incorporated into the host genome, but in case for RNA, there is no chance of that, because RNA, okay, from protein is produced from messenger RNA, and it is done in the cytoplasm. Okay, so there is no chance that this RNA can get integrated into the host genome, because host gene is in the nucleus. Okay, then efficacy. So early clinical trial results indicate that these vaccines generated reliable immune response and are well tolerated by healthy individuals with few side effects. Okay, it's actually was true for when they did the trial for COVID-19 mRNA vaccine and production. Okay, so vaccines can be produced more rapidly in the laboratory in a process that can be standardized, which improves responsiveness to emerging outbreaks. Okay, can be rapidly produced in the laboratory. So such as suppose there is a emergency outbreak, so vaccine can be produced emergency basis rapidly. Okay, now although there are promising things, but still the RNA vaccine, they also have some challenges. So okay, so what are those challenges? Such as unintended effects. So the messenger RNA strand in the vaccine may elicit an unintended immune reactions. Okay, so however, to minimize this, the messenger RNA vaccine sequences are designed to mimic those produced by mammalian cells. It actually also has been done. I mean, this strategy was applied in case of Moderna COVID-19 vaccine. Okay, the sequence of the messenger RNA was modified, so in a way that it will not mimic the mammalian cell messenger RNA. Okay, so I mean to okay, so sorry, actually, I mean, in case of Moderna COVID-19 vaccine, the mRNA sequence was little bit modified. So that's why it mimicked the mammalian cells, the human, I mean, it mimicked the human cells in messenger RNA. This modification is actually necessary because otherwise, so whenever a foreign messenger RNA enter in our body can recognize it's a foreign messenger RNA. So the body will readily try to digest it. Okay, so that's why it is, if I mean modification is possible. So and by those modification, I mean if I mean in case of Moderna vaccine, the modification was done in a way that the human cell, they cannot recognize, I mean, so they cannot recognize that it's a foreign. They recognize it as its own. Okay. Okay, it was challenged regarding unintended effects. Then delivery. Okay, so delivering the vaccine effectively to cells is challenging since free RNA in the body is quickly broken down. RNA, I mean, RNA are not like DNA. DNA are very stable, but RNA, they are actually, they get readily broken down. Okay, they get readily lysed. Okay, so it's, I mean, it's tricky to deal with the RNA. Anyway, to help achieve delivery, the RNA strand is incorporated into larger molecule to help stabilize it and or packed into particles or liposomes. Actually, that was done here. I mean, you have the messenger RNA. Theoretically, that directly could be used as vaccine, but messenger RNA are very unstable. Okay, they get readily digested, so that's why those RNA, they are encoded. Okay, and in this encoding can be done by nanoparticles. So in the Moderna, they use lipid nanoparticles to coat the messenger RNA. Okay. Okay, then storage. I mean, challenges with storage. So many RNA vaccines, like conventional vaccines, need to be frozen or refrigerated. So work is ongoing to reliably produce vaccines that can be stored outside the cold chain. I mean, if we need to store them in frozen or refrigerated, we have to use the cold chain throughout our storage and transportation. Okay, however, I mean, scientists are trying to produce a stable RNA vaccine which can be stored in, such as, for example, room temperature. So cold chain would not be necessary to maintain. Okay, so anyway, we hope that soon this kind of vaccine, I mean, the scientists, they will be able to produce this kind of RNA vaccines which can be stored in the room temperature. Okay, so however, so this was all about the vaccine overview. Thank you for your patience.